In this video, we will discuss about in vivo mutagenesis. So, before going into the topic, let us first quickly understand why there is a need of in vivo mutagenesis. So previously, in vitro mutagenesis is been used to create mutations within the or DNA, or we can say mutation libraries. So what happens in vitro mutagenesis is that there is a lot of time and effort is required. It is a laborsome process, as we need to use error-prone DNA to create the mutation with the help of this PCR reaction, and after that we need to separate out the mutations and amplify it, and then confirm them with a final assay of final detection assay. Whereas in case of in vivo mutagenesis, most of these things will be done by the cell itself. So the uh, approach that is used in this paper, or we can say in this video, is that T7 RNA promoter. It is the fusion of T7 RNAP and PMCDNA1. The function of this T7 RNAP or T7 RNA polymerase is to direct the PMCDNA1 to the site of target, and this PMCDNA1 will then act on the cytosine of the DNA and mutate that cytosine of the DNA. However, there are DNA repair and enzymes are also present, such as MHH6 and APN2, and when they interact with the T7 RNA promoter, then it will create a more different mutations, because instead of repairing it, it interacts with the T7 RNA promoter and works in a way that finally leads to create more different mutations within the DNA. And some of the examples are shown here, in which this MMR, that is mutation, that is MMR is the mutations results from the interaction of DNA repair enzymes. I'm not able to recall the exact full form. So what happens in this case, or what is seen in this case, is that this GU in the DNA is uh, mutated to TA, and in case of error prone mutation, or we can say error prone replication, what happens in this case is that this UT and GA is replaced to CG and GC, and whereas in case of replication, when the mutation is done to replace cytosine to uracil, the in the replication process, what happens is that the UG will replaced by TA, and the whole sequence in this panel B shows the replacement of G with A in a complete gene sequence of a particular protein, and we can see that this orange color represents the mutation when uh, first start with the blue color. Blue color represent the mutation when this T7 RNA promoter is present, and this gray color represent the control lines, whereas this blue color also represent the control lines in which uh, this one of this T7 RNAP or PMCDNA1 is not present. You can refer the paper to find out the exact details. And this is the same figure. This is the mutations, or we can say mutation, the URA3 uh, enzyme is inhibited or is inserted into the cells with a premature stop codon TAA, and in this kind of cell lines, and with the help of this T7 RNA1 promoter, mutation has been carried out or and then finally quantify to check in which of the cell lines this mutation TA mutation is reversed, and the results are shown here. In this MSH6 and APN2 cell lines, results are more prominent, and this MSH6 and APN2 uh, is related to DNA repair enzymes. So we can see that this MH6 and APN2 promotes more mutation when interacts with T7 RNAP and PMC DNA1. So I hope you have understood this, how this in vivo mutagenesis process works in brief. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.